Alright, I'm back. So my computer froze in the middle of the last recording, so it will be in 2 recordings, unfortunately. Well, hopefully it doesn't freeze again, or it will be in 3. But we'll just go right where I left off from before. So the last slide that we talked about was this one. So now we're going to go into DNA replication and passing on genetic material. So duplication is exactly what it sounds like. It's making an identical copy of the genome so that it can be passed on to the new daughter cell or offspring. So it must be completed during a single generation time, so it needs to be fairly quick. And for that reason, there are many enzymes working at any given time doing the replication process. Some cells, DNA replication can happen in a matter of minutes. I know for T cells it only takes about a couple hours, which is pretty nice considering they're a human cell and they have a fairly large genome. Bacterial cells, E. coli can do it in less than a minute. So it's fairly quick, fairly rapid for smaller genomes. This requires the action of 30 different enzymes, which I'm not going to expect you to know all of. There's just a handful that are important for what we're talking about. And the basic concept is to separate the strands. So you have double-stranded DNA. You need to separate them into the two compartments, or the two components, the 5' to 3' sense strand and the 3' to 5' antisense strand. You then copy both of those templates by adding new free nucleotides to both single-stranded pieces, making 2 single-stranded copies of, or 2 double-stranded copies of the DNA. And then this will produce 2 daughter molecules, which then can go, one will go to the daughter cell, one will stay with the parental cell. So a couple of the important enzymes I'll expect you to know. Helicase, so this is for unzipping of the DNA helix. This binds to the A and T rich sites, breaks those double bonds, and then kind of zips along the double-stranded DNA, breaking it up into its 2 separate pieces. Primase, so this adds an RNA primer. This RNA primer is what tells DNA polymerase where to bind. Once DNA polymerase binds, it can then grab free nucleotides that are in the cytosol and add them to the single-stranded piece of DNA. DNA polymerase 3 is the one that adds the bases to the new DNA chain. DNA polymerase 1 removes the primer, so this removes the RNA primer that primase placed, and then it closes the gaps by adding nucleotides in that bit where the primer was bound. Ligase then comes through and closes all of the gaps between the deoxyribose sugars in the sequence. So when DNA polymerase 1 comes through and fills in the gap where the RNA primer was, it just puts in nucleotides, but it doesn't actually make the covalent bonds between the deoxyriboses. So ligase will come through and kind of push them together, creating these nice covalent bonds. And topoimerase 1 and 2, so this is for supercoiling and untangling. So these are kind of larger proteins that will kind of prevent the DNA molecule from kind of folding in on itself, allowing it to be open. And by being open, it allows for DNA polymerase to bind and add in nucleotides. So the most simplest form of replication, which happens with small genomes, so you see this a lot in prokaryotes, is semi-conservative replication. So each strand acts as a template. So here's our double-stranded DNA. We break apart the weak hydrogen bonds with helicase, creating 2 single-stranded pieces of DNA. Then you add in DNA polymerase 3, which binds to the RNA primer that was laid by primase, and it reads in a 5' to 3' direction on both sides. So it kind of works in opposite directions. This works really well if you have a circular genome, because you can kind of start at one end, start at the other end, and then just kind of go zip around until you reach the other end of the circular genome. If you don't have a circular genome like in us, this becomes almost impossible and kind of a waste of energy. So we do something a little bit different and a lot more complex. This allows for accurate replication, but again, it is somewhat slow, especially if you have a large genome. If you have a small genome, it's perfectly fine. The biggest thing to take from this slide is that DNA polymerase III reads only in the 5' to 3' direction. It will never go in a 3' to 5' direction. So for that reason, it has to go this direction, and it has to go this direction on the opposite strands. So it's going from 5' to 3' this way, and then 5' to 3' this way. So the starting bit is the origin of replication. So that's the site where helicase will bind and start the unzipping. So it's a short sequence rich in A's and T's. These A's and T's only have 2 hydrogen bonds, allowing for a more weak interaction. So where helicase can kind of just plow right through. It's held together by only 2 H bonds rather than 3, obviously, and then less energy is required. The helicase binds to the DNA at that origin, untwists the helix, breaks the hydrogen bonds, and results in 2 separate strands of DNA. Here's another video. You can check that out at your leisure. I won't play it because it just wastes memory. So DNA polymerase synthesizes the new daughter strand by basically grabbing free nucleotides, and these nucleotides will bind to their counterpart. So A to T, C to G, and vice versa. The process depends on several other enzymes as well, but key points about DNA polymerase III. So it does require a whole bunch of different factors that kind of bind the DNA polymerase and allow for this process to happen. But for our purposes, I just expect you to know what DNA polymerase III is. So nucleotides that need to be read by DNA polymerase are buried in the double helix. So the DNA must first be unwound and 2 strands separated. So that makes sense. If they're in their double-stranded positioning, those nitrogenous bases are hidden, and it makes it impossible for free nucleotides to bind. DNA polymerase III is then unable to begin synthesizing. That's where helicase comes in and breaks these hydrogen bonds, allowing for DNA polymerase to bind. And it can only add from a 5' to 3' direction. And these nucleotides are regularly synthesized by the cell, or they're brought in from the environment. So there's always free nucleotides kind of floating around for DNA polymerase III to grab a hold of and create these new strands. So as the replication proceeds, new double strand begins. DNA polymerase I then removes the RNA primer that primase laid down, and then replaces it with DNA. Once this happens, like I said, you have these little gaps between the deoxyriboses, so the covalent bonds aren't fully formed yet, and that's where ligase comes in and fuses these together, creating a nice, solid, strong piece of DNA. Oh, now it's going. My computer keeps acting up and freezing, so I don't know what the deal is. Now, this is a little bit more complex. So before we were talking about, basically you have one circular chromosome, like here. And you put one DNA polymerase III here, one DNA polymerase here, and they go in the 5' to 3' direction in opposite directions until you get 2 single-stranded pieces, or 2 double-stranded chromosomes that are copies of each other. If you want to speed up the process, you can put 4 DNA polymerases, so 2 on each single strand, and then they will move in opposite directions. But the problem with this is that you can only read in a 5' to 3' direction. So if my 5' to 3' direction is here, that means this guy can read perfectly fine, but this guy can't, because he can only go in this direction. So he can start moving in this direction, but he'll run into the double-stranded DNA that this DNA polymerase created instead of going in this direction. So a way around this is by using the enzyme primase, and the primase will then travel down this strand and place RNA primers in random locations, about 100 nucleotides or so away from each other. And what this does is say, okay, DNA polymerase III, every time you see an RNA primer, start adding nucleotides. So it can then go down the line in the opposite direction, find an RNA primer, bind, then fill in the bits in between, going in the 5' 3' direction. unbind, go back, find the next RNA, which you can see here. So here you have this guy, he's going in the 5' 3' direction without a problem. But this guy, he can't do that, obviously. Going in this direction is 3' to 5'. It's impossible for DNA polymerase to do that. So these little triangles here represent the RNA primers that were added by RNA primase. And this basically says, okay, DNA polymerase, bind at these RNA primers and then fill in until you reach double-stranded DNA. Go to the next one, fill in until you reach double-stranded DNA, and go for on and on and on and on. That's when DNA polymerase 1 comes in, removes these RNA primers, and adds in nucleotides, but there's still little gaps in between the deoxyriboses, and that's when ligase comes in, fuses these deoxyribose gaps, allowing for a strong segment of double-stranded DNA. Here you can see this is what happens as the DNA is being copied. You get these two identical copies of the circular chromosome, and at the very end you get something called a nick, and this little nick eventually breaks off, allowing for two separate copies of DNA. Here's another movie you can watch at your leisure. Don't worry about that. Alright, so applications of the DNA code, so transcription and translation. So now we've made copies of our DNA, meaning we can make copies of ourselves. But the important, or the most important, part of the cell life cycle is producing proteins. If you don't produce proteins, you don't survive. So the central dogma behind all of this is taking DNA, making an mRNA copy of a gene, and then taking that mRNA copy and translating it into a protein. So to go from DNA to RNA is called transcription. You've heard me talk about this before, so it should be just review. Then going from mRNA to actual protein is translation. Transcription is fairly easy to understand, and that will be a real easy, quick talk point for me. Translation is a little bit more complex, and we'll probably have to go over that when I get back on the board, just because it makes it a little easier to see or to understand when you can see it. Some exceptions to this obviously are RNA viruses. So they have their genome is actually RNA instead of DNA. So they obviously don't have to go from DNA to RNA since they're already RNA. Okay. In fact, some retroviruses will go from RNA to DNA and then from DNA to RNA. So they're a little different. So in addition to RNA that produces proteins, other RNAs are used to regulate gene function. These are called shRNAs. They're just small RNA fragments that actually kind of interfere with translation, and they're used as a way for the cell to control how much protein is being produced. We also utilize them in the lab to shut proteins down and then see what happens to the cell after that protein is gone. And then many of the genetic malfunctions that cause human disease are found in these regulatory RNA segments. So here's just kind of a picture of the central dogma. You have your DNA, you transcribe it into mRNA or other versions of RNA. tRNA is what aids in translation. rRNA is what makes up the ribosome. But most of the time it's transcribed into mRNA. mRNA in a eukaryote will then translocate outside of the nucleus, where it can then be translated via a ribosome. If you're in a prokaryotic cell, this all happens almost simultaneously. You'll get your mRNA transcript, then you'll translate it right in the same spot as the DNA, because there's no nucleus, obviously. During translation, you're adding on amino acids until you form a nice amino acid strand called a peptide. And these peptides get longer and longer and longer, and then they kind of get folded by different cellular machinery like the Golgi and endoplasmic reticulum until you get your final product of a protein. So gene-protein connection, so the triplet code and the relationship to proteins. So 3 consecutive bases of DNA strands are called triplets. These triplets code for an amino acid. There is redundancy in the code, so multiple triplet codes, or codons as they're called, can code for one amino acid. So this allows for mutations. This prevents mutations from having a super great effect all the time. That's because if you just do a point mutation where you just change one amino acid, the chances of you changing it so that it becomes a totally different amino acid are reduced by 3, essentially. It's transcribed and translated, and it dictates the type and order of amino acids in a polypeptide chain. Proteins ultimately determine phenotype, and DNA is mainly a blueprint. Like we've mentioned before, it's the basis of, it's the code, it's what describes everything. So here we have DNA, it's being transcribed into mRNA, then we have amino acids during translation that each one of these amino acids code for 3 nucleotides, or 3 nucleotides code for 1 amino acid. And these 3 nucleotide segments are called codons, and there's some redundancy built into these. So RNA is essentially almost the same thing as DNA, except instead of a deoxyribose sugar backbone, it has a ribose sugar backbone. Can be in helical form, can be in single-stranded form. It doesn't have T or thymine, it has uracils. So instead of AC, T, and G, it has AC, U, and G. And the sugar is ribose, obviously. A transcript of a structural gene, that's what mRNA is. It's basically a RNA copy of the DNA. It's as simple as that. The reason we do this is because A, we want control over what's going to be produced. Having an mRNA message is one way of controlling this, especially in eukaryotic cells. Just because we produce an mRNA message doesn't mean it's going to get translated. It first has to be translocated out of the nucleus before it becomes translated, so it's a way of controlling. Also, mRNA isn't forever. It is degraded. So if it gets degraded at a fairly regular rate, then we don't have to worry about too much of that protein being expressed. Also, your DNA is very special. You don't want it to be constantly played with or manipulated, and that is what would happen if we used it to directly translate to protein. You want a filler message in between there that isn't that special. If mRNA gets degraded, not a big deal, we'll just make more. If the genome got degraded, there's nowhere to go. You can't make more. It's a problem. So transfer RNA or tRNA, so this is a 75 to 95 nucleotide long length piece of RNA, and this RNA has something called an anticodon built into it, and the anticodon is what binds to the codon of the mRNA. And so this anticodon will bind to a specific codon, and attached to the other side of the tRNA is an amino acid. So this is what allows for the tRNA to bind to the mRNA, putting the amino acids close to each other, allowing them to bind together, forming a peptide or a piece of a protein. So it's where the T comes from. It actually transfers amino acids from the cytosol to a ribosome and allow them to bind to their corresponding codon sequence on an mRNA segment. And there are 20 amino acids, and 20 amino acids, different combinations of those are what create different proteins. So here are the different RNA molecules. mRNA, you can see it just looks like a single-stranded piece of, or single-stranded molecule of nucleic acids. And then transfer RNA has got this kind of secondary structure. It looks like a T. You can see the anticodon here. This is what binds the codon of the mRNA. And then here you have the amino acid attached to it. So during translation, this guy will come up, bind to its corresponding codon, and its amino acid will then bind to a neighboring amino acid that is being bound to this codon. And by this happening over and over and over and over and over again, you get a nice linear amino acid polypeptide. So the ribosome, it's made up of rRNA. It's essentially got 3 different sites, the EPA sites. It's pretty easy to remember, Environmental Protection Agency. You need something to help you remember it. Prokaryotic 70S ribosome, so it's composed of tightly packed rRNA. It's a little smaller than the eukaryotic ribosome, and it's definitely different, and this is a good target for a lot of antibiotics. So the RNA component is a long polynucleotide molecule, but it's very, very large. But again, it has to be made out of RNA, because if the ribosome was made out of protein and the ribosome is the only thing that can make protein, it obviously can't make itself, so you run into problems. And the interactions of the protein and RNA create the two subunits of the ribosome. That engages in the final translation of genetic code. So you have two subunits, the large subunit and the small subunit. The mRNA segment right here will kind of bind in between. And then you have your tRNAs binding with their anticodons, binding to the codons. And this puts two amino acids in close proximity. These amino acids can then bind, creating a polypeptide. And then what ends up happening is this guy gets ratcheted down so that this site will then move down here, this site will then move down here, and then you'll have this site here in this new A site, allowing for a new tRNA to bind. So it's basically like a chain reaction that occurs. So transcription is fairly easy. You have RNA polymerase II. It binds to the double-stranded DNA, it unzips the double-stranded DNA, and it adds free-floating RNA nucleotides, creating an mRNA transcript. Now there are other enzymes that do aid in this, but for our purposes, I just expect you to know RNA polymerase II. And he kind of does everything. He binds, he unzips, he adds in free new RNA nucleotides, creating the mRNA transcript. It's as simple as that. Here's a movie. Translation. Now this is the second stage of gene expression. This is where you're creating a protein from the mRNA transcript. So all the elements need to be synthesized. Proteins are brought together by the ribosome. There's 5 stages: initiation, elongation, termination, protein folding, and then protein processing.